Hello and welcome back to our series. We are grateful you're here. We are always expanding our educational opportunities, including our growing collection of programming led by Drs. Armin Smith and Jeannie Hoffman-Sensis, and program team members Pamela Getz, Samantha Rockler, and Stephanie Gruber Greenberg, specifically tailored around the experiences of women with bladder cancer. See our website for the next virtual women's group. Additionally, we have been working with our clinical providers to assure our GBCI front door for care is easy to access. We have two email addresses, bladdercancer@jhmi.edu, and a more specific email for upper tract cancer patients, utuc@jhmi.edu. Before we introduce today's session, I wanted to let you know that you can find our updated programming and useful resources on the web, including a new one-page bladder cancer patient navigation aid designed to help our patients understand the complex relationships between our unique teams and departments across the GBCI. You can download a copy from our website landing page or email us at bladdercancer@jhmi.edu to request this valuable tool. While on our site, I encourage you to take a look at our previous Grand Round series that are all viewable through YouTube. Please also mark your calendars to join us next month, March 19th, for a panel discussion with Drs. Noah Hahn and Max Cates, who lead our new non-muscle invasive bladder cancer program. Hear from them about our work to stand up a novel multidisciplinary care model focused on NMIBC. I'm looking forward to an engaging discussion. Again, more information for all of our programming can be found on our website. Just Google Greenberg Bladder Cancer Institute. And if we can help you directly, we are only an email away at bladdercancer@jhmi.edu. I'm grateful to all of you for joining us today for a conversation with our co-founder, Stephanie Gruber Greenberg, and members of the Greenberg Bladder Cancer Institute Laboratory. In a moment, I will ask our lab team, Stan, Stefan, Chisom, Alina, and Kai, to introduce themselves to you. But first, I would start with a brief overview of some of the projects inside our portfolio that you'll hear more about today. So just the order of speeches that you'll hear today will be Stefan, Alina, Chisom, Kai, and then finally Stan. So I'm going to go ahead and share my screen. So we're going to take you on a little tour, partial tour of the research that's going on in the laboratory. So for those of you who are ever interested in visiting us, feel free to come on by. We are happy to give you a tour. So we're located in the Park building on the second floor, and we have an open laboratory space. I think Stan's going to share some pictures with you. Our laboratory is fully equipped for a lot of different things, including what you may have heard of called next generation sequencing. And our group has growing data science capacity in collaboration with the Brady Urological Institute. You'll hear about data science today from Kai. So we have a very multidisciplinary group of principal investigators. We have 3 medical oncologists. I know you know who they are. We have 4 urologists, and now we have 3 data scientists. So our preference is to focus on problems that are directly clinically relevant. So what we like to do is we like to ask questions based on things that we see in real patients and their tumors. And some of the questions that we're interested in answering right now, and this is just some of them, include, why do some bladder cancers metastasize? We know that metastatic disease is what kills people, but it's still not well understood why they do that. So a very important part of our research, our collaborative work, is to try to understand why some bladder cancers metastasize, so that we can understand those mechanisms and target them with new treatments. Another question that I think you've heard me talk about before is, can we use measurements of urine tumor DNA to better track bladder cancer? We've heard from a lot of you that you don't like cystoscopies. They're kind of a necessary thing right now, but we can see a time when they'll be informed by having more objective measurements of urine tumor DNA in the urine that can tell us things about that cancer that may or may not be visible by cystoscopy. Another question that we'd like to ask is, can we use computers to see subsets of disease that aren't yet visible to us? You've probably all heard of artificial intelligence and machine learning. These tools may help us see subsets of disease that we can't see looking at one thing at a time with the limitations of our human brains. And then finally, can we identify new therapeutic targets that can increase the number of patients who will benefit from immunotherapy? Immunotherapy has changed disease courses, both for patients with advanced disease and also with non-muscle invasive disease, but it doesn't benefit everyone. And so we're performing research to understand how we can expand the efficacy of these agents so that they can help a lot more people. So with regard to the mechanisms of metastasis, you'll hear from Stefan today. He's working with another urologist named Yujiro Hayashi, who's from Osaka University in Japan. And they're currently using next-generation sequencing on upper tract tumors to see what's going on between the tumor itself and the metastases. And our friend Noah Hahn is about to do the same thing for lower tract bladder cancers. And finally, we've established collaborations both with teams up in New York and also a new one that we've established with a group at Purdue, where we're actually going to compare what we see in human beings to what the Purdue group sees in dogs. And this project will involve a lot of data science. So you may have heard about liquid biopsies before. Most of the time, these refer to measurements we're doing in blood to measure tumor DNA in blood. We think those measurements in patients with bladder cancer are probably telling us more about metastatic disease. Whereas in urine, we think we can use the same kinds of approaches to understand what's going on in the bladder. So we've established several collaborative projects with a company that's based in San Francisco. Stefan is leading the effort with upper tract cancers. And they've developed a means of sequencing tumor DNA in the urine that's called UroAmp. And the pattern of mutations that's present tells us about how aggressive the cancer is, and the amount of DNA that we measure can tell us how much cancer is there. And so we think that this or another similar test, because there are many in development, could be very useful in surveillance, what we currently do with cystoscopy, and also to measure the effects of our therapies, things like BCG, gemdosi, and some of the newer agents like adstiladrin and others that you've heard about. So we're very excited about urine liquid biopsies, and we're very excited about this collaboration we've established. So Alina is going to tell you about this drug, enfortumab vedotin. Probably a lot of you have heard about this drug already. Alina is going to give you more detail today. So Alina and Jeannie made an important discovery, which was in the patients who develop a toxicity in their skin, they seem to have much better clinical outcomes. And so the team is now working with another group at Johns Hopkins to identify the mechanisms involved. We think that these mechanisms may provide us with biomarkers that can tell us very early on who's going to benefit from this very exciting drug. So Chisom, we recruited her to be our data coordinator. She's a clinical data scientist. And what we knew going into the GBCI from work that we've done in clinical trials is the best data that we have are collected very intentionally. Clinical trials, you have to spell out the rules in advance, how you're going to collect clinical data, and you do it in real time. And so we wanted to do the same thing here at home for our patients who are just receiving conventional real-world care for urothelial bladder cancers. And so Chisom is helping us offer this opportunity to patients who want to participate in our research. So she does something called consenting, which means she sits down with patients and tells them about our research and asks them if they'd be willing to participate in it. In addition to that, she's also learning to help us perform DNA sequencing on their tumors. And so the plan is to get all of these data together in the bank right at the very beginning. So when we know things are the most robust and rigorous. And this will serve as the basis for all the other research that we do that you'll hear about over the course of the next few years. So modern cancer research relies heavily on something called data science. So just to put it in kind of a simplistic term, data science is essentially using computers to analyze very large data sets. So the data sets we generate from our sequencing, for example, are very, very large, and they require specific computer programs to help us analyze them. So Kai is amazing. He's a self-taught expert in data science who's actually been very instrumental in helping mentor other members of our group. And while his current research is focused mostly on understanding how the bladder cancer drug erdafitinib affects the way the immune system reacts to bladder cancers, he's doing a lot of other work as well. I think he's going to give you an overview of what data science is, and he'll be available to answer questions you have about it. And finally, all of the work we do is dependent on the generosity of our patients who give us permission to use their clinical data, but also permission to use their tissues. And when I say tissue, I mean tumor tissues, but also urine and blood. So when tumors are removed from patients, their bladder cancers are sent to pathology for review. This is very important for clinical planning. So the pathologist provides us with staging information, grading information, whether or not there's a variant histology present. These are very important prognostic tools for urologists and medical oncologists to create their clinical plans. But there's always, for the most part, tissue left over, and it's preserved by fixing it in a substance called formaldehyde, which preserves it, and then it's put into wax blocks and stored. So these blocks are cut into very thin sections, and these very thin sections are used for diagnostic purposes. But we can also use them in the laboratory to do things like DNA sequencing. So we were very lucky to recruit Stan to the group because he came to us from pathology. He knows the ropes over there, and he helps us obtain these samples from pathology, and he also helps to stain them, to measure things that we want to measure in these tissues. And he's our lab manager as well, so he's kind of the ringleader of everything we do. He does a lot more than just do this tissue-based work, but we're very lucky that he has this good experience and he brings it to our group. So that's it for my introduction. Now maybe we can go around and each of you can introduce yourselves. Let's start with Stefan.
Hey, I hope you can all see me. My name is Stefan Brünemann. I'm a urologist from Austria. I studied medicine in Austria, did my residency here at the Medical University of Vienna, and are now in my 6th year of training. And I use this year to do a year of research fellowship at the GBCI in Baltimore. Yeah, my interest clearly is uro-oncology, and at the GBCI I focus mostly on UTUC. And I think, or I want to talk about like specifically 2 projects that have both been already introduced by Professor McConkey. One is about lymph node metastasis in UTUC, so upper tract urothelial cancer. And the other one is the collaboration with Convergent, a company from San Francisco, where we look at biomarkers and non-invasive diagnostics. So the first project I want to talk about is the lymph node project, because we try to understand in this project what makes cancer cells spread to the lymph nodes. So that's usually the first place where they spread, if they spread. So it's like the regional lymph nodes that surround the kidney. And we really want to understand why some tumors metastasize really early and others not. And the big plan behind this is to really avoid over and under treatment in this patient population. Because like, as you've heard, there are like new treatment therapies upcoming, like systemic therapies, but also different kind of surgical treatments, like endourological treatments using lasers for less aggressive tumors, and more aggressive tumors need a more aggressive approach, mostly meaning like removing the whole kidney and the ureter. But in UTC, it's even more complicated than in bladder cancer because tissue sampling is quite difficult. You have to go up the whole ureter with a tiny scope, and then you get like usually a tiny piece of tissue, and the pathologist try to make the best out of it. But risk stratification is really difficult for this reason, and also using imaging, it's not really clear how invasive, so how deep this cancer grows into the mucosa or the kidney itself. And therefore, we try to identify mutational signature of those cancers to really improve risk stratification and tailor the right treatment or the ideal treatment, and avoiding over-treatment, but also under-treatment. So how does it work? We first identified patients from our database. So we have a retrospective database of patients where we have like different like parameters like about the patient, like name, birth, or actually not the name, it's de-identified. So every patient has just a number in there, date of birth, the clinical staging, the pathological staging, and other important information. And we go into this database, look through all the patients and identify or identified patients that had a UTUC, so upper tract urothelial cancer, and also lymph node metastasis. Then we went through the pathology reports to order the blocks and the slides. So what is a block? A block is a piece of tissue embedded in paraffin wax. And a slide is just a glass slide with a thin cut of this slide that usually gets stained, and where you can look under the microscope and identify the cancer cells. So we ordered all the blocks and the slides of those patients and tried to cut new slides, which we can use for DNA and RNA isolation, and finally sequencing to really identify those mutations. And it's really a process, because like sometimes you don't get the blocks you want, you don't get the slides you want, you have to go back to the pathology report or the new blocks, see the pathologist again to identify new slides and cut new slides from those blocks, which is like a bit of a bottleneck. And then also the problem is the tissue doesn't get better with time. So we went back till 2004 and saw that the RNA, especially the RNA, is quite fragile and disintegrates in the archive quite quickly. But we're also like thinking of new ways to improve our biorepository, to improve like ultimately our research and research results. So currently we're in the process of isolating DNA and RNA, even like new patient slides are coming in right now, and we just started to sequence and analyze the first matched pairs. So this is quite an exciting project. And another exciting project is one with Convergent, where we try to identify urinary biomarkers, because we also run into issues in diagnosing, as you heard, like UTC, especially considering cytology. So cytology is the current approach we use in the everyday clinical practice, together with cystoscopy. So we'll look into the bladder, take a tiny portion of urine, send it to the pathologist, and they look under the microscope and just describe how those cells look like. So it's a purely morphological description of those cells, and then they can say like, it looks suspicious, or it doesn't look suspicious, or sometimes they say they cannot really tell. It might be suspicious, but they're not 100% sure. And this approach works quite well in bladder cancer, but in upper tract urothelial cancer, it doesn't work that well. And also like in low-grade disease, so in less aggressive tumors, it also doesn't work well. So therefore, we tried to improve that together with Convergent. They started with a gene panel in bladder cancer, which has already been validated and also published. And now we try to achieve the same in UTUC. So therefore, we collected urine from 41 patients just before they got the operations, and also like at multiple time points after that, and analyzed the DNA out of it. Considering this approach, problems you can run into is that also like urine is quite hostile, so it disintegrates the DNA quite quickly. But Convergent fortunately developed kind of like a special fluid, like a preservative, to save the DNA, like in order to avoid this disintegration. And they were very successful with it, and showed that this works for up to 14 days, and it doesn't, like, we don't need to freeze it. We can like store it at room temperature, and this is really great because it's it's a very smooth process. If you don't have to freeze it, you can just ship it, and you have enough time to even send it to the West Coast. So we identified 41 patients, and we already did the first analysis. We correlated it like in the first place with the pathological staging, and we were able to get conclusive results of every single patient. So it was like 100% of the patients could be genomically profiled. And we were also like successful in predicting the grade, which was also like grade prediction, meaning like how aggressive is this tumor, was also like 100%. So we were able to predict the grade in every single patient. Then there was one patient who received neoadjuvant chemotherapy. He responded really well to it, so there was no tumor left in the kidney. And this test could also predict that, which is really important information to us. And it also was significantly better than cytology. So this also brings us back to precision medicine and trying to avoid too invasive diagnostics, because as Professor McConkey already said, surveillance is really important in those patients. Predicting disease recurrence or detecting disease recurrence is really important to be able to salvage those patients, and also predicting the response to those new treatments is really important. So far, we mostly use CT scans, where you always have like a bit of a time shift, and like really, you're like it's also like a basically morphological description of the pictures you take, and also yeah, we want to be able to predict or detect disease recurrence as early as possible using.
Stefan, I'm going to interrupt you for a second.
Sure.
So in the first project with the lymph nodes and the tumor, even though the tumor is hard to find, thank you for chasing it down as hard as it is. You're looking for a match so that the lymph node and the tumor are saying the same thing and telling you something important. Is that correct?
Well, yes. Yeah, we look for basically drivers of metastasis. So we look like at the malignant cells in the lymph nodes, and in the primary tumor. And then we will compare and see like if there are like certain cells in the primary that metastasized to the lymph nodes, and if there's like something in common, and if we're able to
That's
predict
super important information for treatment. Like it can tell you a lot because it's sort of validated in 2 places. Is that correct?
Well, yeah, basically go back. We want to predict, like if we have like, for example, a patient with a UTC, upper tract tumor, how high his chances are that he has already developed or will develop lymph node metastasis, and how aggressive we have to treat him.
That's important information. Wow. And this is really exciting. Tumor, I mean, urine, our urine holds many messages, and you're trying to unlock the secrets of what that urine holds so that it can better inform treatment. And it seems to be moving along very quickly, the biomarker development. I know David McConkey has things to say about that, but thank you for this. This is fascinating. And it's very positive. It's more than has ever been there before. That's for certain. We can't acknowledge how hard it is to do the things you do with the wrong slides or the urine goes. I mean, we have no idea how hard the work you do is, but we thank you for it. And it seems overly like a real sort of step in the right direction to help people understand, and physicians and clinicians understand like what's really happening by digging deep. So, wow, thank you for that. David, I hand it back to you. I'm really
Yeah, so the other thing that Stefan didn't say is that he's sitting in Vienna.
I am.
So thank you for joining us a little bit later in the evening than it is here.
And Stefan, one thing, you are a urologist by training in Austria already, and you've come to the United States your 6th year to do research. Is that?
Yeah.
And then you returned to Austria to be a physician, clinician scientist in Austria. Is that right?
Yeah, that's the
If
plan.
he ever let you go.
Yeah, well, we'll get the next one, and we'll keep working with him when he's there. So
What he won't tell you is he was deciding, I heard a different conversation. He's a world-class cellist, and he was deciding between music and medicine. So thank you for choosing medicine, playing the cello on the side. He went to music school and then he went to medical school. Like, fantastic. Thank you, Stefan. Stay with us. We probably will have some questions. And if anyone has questions out there, please put them in the chat and we'll answer them. And I hope this is fascinating because I think it is. And these are the young people who are making this happen. Toiling in obscurity, no longer.
Yeah, hopefully not so much. So Alina is definitely going to be interesting to you. Alina is another international member of our lab. Go ahead, Alina, tell us about yourself.
So thank you. Hi, everyone. I hope you can see me. My name is Elina Vlachou, and I'm an MD, originally from Greece. So I graduated from the Medical School of University of Patras in Greece in 2022. But I actually first came here even before I graduated. I was a medical student and I was doing some rotations here at Hopkins. And this is how I met actually Dr. Hoffman Sensitz and started working on a really, really cool idea he had that's actually, it's grown and it's developed into the project that I will be talking to you about today. So after I graduated, I came back and now I am working as a postdoctoral research fellow with GBCI. Our project has to do with the chemotherapy drug that I think many of you have heard of. It's called enfortumab vedotin, or EV, and it's what we call an antibody drug conjugate. It's FDA approved for advanced urothelial cancer. And so when we say it's an antibody drug conjugate, what we really mean, it's a smart form of chemotherapy. It consists of the chemotherapy molecules, and they are attached to an antibody. And what this antibody does is it targets cancer cells throughout the body. And that way it helps deliver the chemotherapy agent directly to the cancer. Response rate for EV when it's given by itself is 40 to 50%, meaning that of the people who receive the treatment, we see the tumor shrinking on the scans in 40 to 50% of patients. When it's given in combination with immunotherapy, an agent called pembrolizumab, response rates are even higher, to about 68%. And very recently, a phase 3 study compared EV with pembrolizumab to the traditional cisplatin-based chemotherapy, which had been the standard of care for decades. And this study actually showed that EV with pembrolizumab had higher response rates and longer survival. And that led to its FDA approval this year for first line in advanced urothelial cancer. So one of the most common side effects we see with EV is some skin reactions. This could be rash, it could be itching, it could be darkening, could be rash with itching, and rarely we see some people having more severe reactions to the point that they may need to receive some oral steroids or even be hospitalized to be supported. So what our project has to do is with this observation that skin reactions are actually associated with improved patient outcomes. So we looked at patients who received enfortumab alone for treatment of advanced urothelial cancer here at Hopkins. And much like Stefan described, we created a database that included around 90 patients. What we saw by analyzing those data was that people treated on EV who experienced these skin reactions had actually improved survival, and their tumor had higher response rates compared to patients that didn't have these events. Something that I find very interesting is that in our dataset, our group of patients that we looked into, all of those who had a complete response, meaning that their tumor kind of melted away and was no longer visible on the scans, they all experienced some kind of skin reaction while they were on EV. And what was also very interesting and very promising was that the overall survival was also longer among patients who experienced these EV-related events. So what this study is retrospective, and meaning that we looked back and saw patients that we treated in the past and analyzed those data. So this is a hypothesis generating work, meaning it's now it's an observation or a hypothesis, and we still need to prove it. And for that, what we're doing now is we have an ongoing prospective study here at Hopkins where we follow patients from the beginning of their treatment with EV, either EV alone or EV with pembrolizumab. And so we follow those patients all the way through their treatment in order to collect their data more intentionally, as Dr. McConkey said, prospectively, and prove this, confirm this observation. And the other thing we do is we collect blood samples from various time points during the course of treatment in order to look for potential markers of toxicity or response, and identify those patients that are most likely to benefit from EV.
Wow.
Thanks, Selena.
Wow, so if you get a rash and you're on, that's good, even though having a rash is awful, but it's maybe sort of well, an analogy maybe not appropriate is when we all got our COVID shots, if we got really sick, we thought, oh, maybe it's really working. So something is going on in the immune system that causes rashes and maybe is hopefully, we love the word melting away tumors around here. Love that. And that's pretty seismic. So this is the start of, from what I understand, a very significant study and a potentially very, very, very significant maybe change in practice. The fact that EV and Pembro may replace chemotherapy is seismic for people. And a rash is not a fun thing to have, but going through chemotherapy is a whole different animal that's really difficult. So, wow, all the way from Greece. Thank you. This is amazing. And how you'll be studying this, I presume, for years and years to come. And where did it go from here? You're just collecting more and more information as time. First of all, how long are people on these drugs for? Is it months or years? How long is the length of these studies typically a patient would be having these infusions?
So patient starts on the treatment, and then they continue to be on treatment unless we have some indication that it stopped working, meaning we see in the scan that the tumor is growing back, or they have some toxicity that is severe, and so they can no longer be treated with a specific agent. So it's really difficult to tell how long someone will be on treatment.
I have a really sophisticated, wonderful question from our friend Karen Sachs. Is the skin reaction an activation of mast cells?
So that's the interesting thing about research is we don't know what it is yet. And to also answer your question, I think it's complementary here. We don't know how long we're going to do this for. What we are doing is we collect data and we analyze them until we have the answer. And this could be next week. It could be in 10 years.
Right, right. Well, Elena, please keep going. And we're just blessed to have you and we don't think we ever want to let you go back to Greece. So stick around.
Well, thank you so much for having me.
And continue to do such great, brilliant work. Thank you very much.
Thank you.
So we're going to turn now to Chisom. You've heard about the importance of prospectively collecting clinical data. Chisom, give us a little bit more insight into that process.
Sure. So just to introduce myself, my name is Chisom Okereke. I'm originally from Maryland. I actually did my undergrad here at Hopkins. I studied public health. And then I took a little detour and I became a 7th grade math teacher with AmeriCorps for 2 years. And then after that, I pursued a master's in public health with a concentration in global epidemiology at Emory. At no point along my journey did I do anything with bladder cancer until I came here. But it's honestly been so interesting to learn more about what bladder cancer is and just the research that's going on around it. And so I've just loved being a part of that. A little bit about what I do. My role is very 50-50, a 50-50 split between wet lab work and dry lab and patient-facing work. And so when I say wet lab work, I mean work that is centered around DNA sequencing and more of the hands-on specimen work. And then when I say dry lab, I'm referring more to the computer-based data collection and analysis work that I do. And so as Dr. McConkey had already mentioned, I also consent our patients to our studies because when it comes to research, we want to proceed with our inquiries knowing that the involved patient data is collected with the explicit informed consent of the patient. We never want to do anything that keeps any of our patients in the dark. And so I really love this aspect of my project because in research, it's honestly such a beautiful and meaningful thing to me, and I'm sure to most of my colleagues, to come face to face with the people that inspire and motivate the work that we do. And it also gives me that opportunity to sit down and have that face-to-face conversation with our patients where I can tell them about what we can do and allow them to ask any questions that they have. I think that research kind of seems like this thing that's just out there. And if you're not doing research, then you're not going to understand it. But I think having that face-to-face conversation makes patients realize like, this is something that I can understand, I can be involved with, and can be something that interests everybody. And so by consenting our patients to our studies, I also expand the access to clinical data that we have, because the clinical data, as Dr. McConkey mentioned, is super foundational to a lot of the work that we do. So once I'm able to obtain permission from our patients, it allows me to go into the electronic medical record, in this case it's Epic, and collect relevant patient data points. And then I take that information and put it into something called a REDCap database, which basically just allows us to have one specific space where we keep all of our data organized. And we can either go back in time and collect data from past patients, which is called retrospective, or we can be collecting data as time goes on, and that's called prospective data collection. I then combine this data, the clinical data that I've collected from the medical record, and from my dry lab work, if you will, and then I combine that with some of the wet lab work that I do, primarily the DNA sequencing that I have been learning of various patient tissues like urine, blood, and tumor. And so, for instance, one of the main things that I'm doing right now is a specific method called survival analysis, and I use something called Kaplan-Meier curves to do that. And so a Kaplan-Meier curve is basically a really informative visual representation of the relationship between some sort of clinical variable or genetic variable like tumor stage or chemotherapy regimen or a specific genetic mutation. It shows the relationship between something like that and an outcome of interest that could be like recurrence or downstaging. And it allows us to have that visual for what is going on between these two different variables. And it's really been informative for our research. And not only does it give us answers, but it also helps us see where else we might want to go next. So yeah, that's kind of an overview of what I do.
Beautiful, well put.
Thanks.
So following Chisom will be Kai, who's going to tell us about data science.
Hi, I hope you guys can all see and hear me. My name is Kai. I'm a 6th-year pharmacology PhD candidate in Dr. McConkey's lab, and I work on a variety of projects. So one of the projects that I work on is helping Dr. Rusty Johnson, who you might have already seen a talk from, and Dr. Yujiro Hayashi, who was mentioned previously by David. But I help with the mouse models that they're creating that will help us understand things like how different drugs can treat tumors and how the immune system interacts with the tumor, something that's very important when studying things like immunotherapy. And this work can help us answer questions like, who is this drug most effective for, or how is the drug working, or can we make this drug more effective? So the way that I answer those questions is by doing bioinformatics. So bioinformatics involves with working with a lot of data. So data that are so large it would be difficult or likely impossible to work with by hand in a reasonable amount of time. And I work with it by writing code, computer code, in a language called R. And this allows me to answer questions from sequencing data from these mouse models and these data sets that contain easily over 100,000 different numbers in them. So I write code that allows me to ask, for instance, what genes are more expressed when a mouse gets a certain treatment? And because we look at nearly every gene that is expressed in the genome, we can make new, surprising, and exciting discoveries instead of limiting ourselves to just a few select genes. This huge amount of data allows us to make some guesses even to why certain genes might be more expressed. So there are other modalities like newer, more powerful methods of sequencing that allow us to look at expression of genes not only in the whole tumor, but inside individual cells inside of the tumor. So as an analogy, imagine as you eat a meal, you can guess what the ingredients are by tasting it. But this new method called single cell sequencing is kind of like getting the recipe. So put maybe a little bit more concretely, this would allow us to know if a tumor has more immune cells in it and even what kind of immune cells those are. So with a mouse model, this would allow us to find things out, like asking if certain treatment is more effective if a certain cell type is present, which could possibly give us potential targets to improve the efficacy of a given treatment. Another method that we can
I'm
use
going to interrupt you for a second. This is fascinating. So the word mice like makes my skin crawl, but do you give the mice these tumors? Do you like make them have bladder cancer? Is that I know that's a really stupid question, but and how did the mice get the thing that you want to study? And how similar are mouse models to human models? Is there parity there?
Yeah, so there are several ways of going about doing it. Sometimes we do something where it's called BDN. So you actually put a drug as analog of cigarette smoke into the drinking water, and then the mice get tumors in their bladder. So that's one way that they can get bladder cancer. And so at least by means of mechanism, you can imagine that that's more similar to what happens with human tumors. There are some differences, particularly in the differences in subtypes. So these BBN models, they tend to be more basal, whereas human tumors, you have a lot of luminal tumors as well. So that's one way. We can also take human cell lines and put them into the mouse, or you can have mouse cell lines and put them into the mouse as well. So how much they reflect human tumor can vary. And something that we're actually trying to do, mostly due to the great efforts of Rusty and Yujiro, is working with a cell line that will allow us to actually study how the immune system works with or against the tumor inside the mouse. So a lot of times when you use human tissue into a mouse, you can't put it into a mouse that has a competent immune system, because the immune system would actually attack the tumor. And so if you want to be able to see more how this would work in a human that has an immune system, you need the right model for that. And so I think as we iterate on these models, getting something that's closer to what is going on in human biology is incredibly important, especially for things like immunotherapy. Does that answer the question?
Yeah, perfectly. Yes, thank you.
Yeah.
And makes mice less creepy. They are now our friends. In crime.
None of us are happy about the fact that we have to work with mice, but there's really no
Right.
other alternative. So
yeah. So just continuing on the different modalities that we're able to use, we have another one that allows us to look at the expression of a wide array of genes across space. So a tumor isn't always the same in every part of it. The edges might be quite different from the center. So while the edges of the tumor are only a small part of many tumors, they're likely to be the most important part in a lot of these because they form the invasive edge. And because of that, it's useful to know how to combat that portion, doing the most dangerous work, which might not reflect the tumor as a whole. So just in summary, we use all of these methods to look at both patient and mouse samples to help us understand bladder cancer with the ultimate goal of improving patient care.
Wow.
thanks, Kai. So last but certainly not least, our lab manager and pathology expert, Stan. Hi, Stan.
Hi, David. First off, remember, questions are encouraged. This is a good learning opportunity for the patient, so feel free to ask us. We enjoy questions. And then also, thank you to the patients. Without your consent and, specimen and tissues, we really couldn't do what we do. So, yeah, thank you. My name is Stanley Rapay. I am the lab manager for the GBCI. I graduated college in 2014 with a BA in German and biology. I started at Hopkins in 2018 over in outpatient dermatology, and I moved over here it's almost about to start my third year. But yeah, I've have a little over half a decade of pathology experience. As I said, I'm the lab manager, official orderer, stockroom stand, pizza getter, whatever needs to get done, I kind of figure it out. But I'm going to talk about pathology real quick. Let me share my screen. Let's see if this works.
Yeah.
Oh,
This is great.
perfect. Okay, so yeah, I wanted to sorry, going through my notes so I don't forget anything. Yeah, so before I go into pathology, I just wanted to show this is the chef and the sequencer. It's what we do for NGS or next genome sequencing. This is a typical lab bench. This is some of us with our lab coats and making biohazard boxes. And then I actually have a little video that we can watch of the chef if it plays. Doesn't look like it's starting. Oh well. It's very interesting, but yeah, feel free to come by and yeah, we can show you. But yeah, for pathology, like everyone said, the tissue's put into a block, it's cut really thin, it's put on a microscope slide. So on the side is an example of a mouse bladder. And these are all the different slides that we look at. And let's see if this works. Hey, it did. So this is a scanned H&E of that block. And this is really zoomed far out. You can see in the corner, it kind of shows you what exactly is being looked at of the screen. But one of the reasons why it's important for us to scan these in is, as you can see with this is just 10x looking at this exact piece, and the square got so small. So these are just a more zoomed in part. And we actually go even further to just look at, the cells, what's going on with them. And, we can figure out a variety of things just from this basic H&E stain, which is kind of the typical for everything, just based on what the cells look like, what the nucleus looks like, whether it looks how it naturally looks, whether there's a huge cluster. Yeah, so one of the things that is currently going on that everyone's talking about is AI, and it's no different for pathology. Some of these things are not easy for pathologists to look at, and it could be really close to half the people might say it's one call, the other half might say it's another. So having these scanned H&Es and using machine learning to have an AI to be able to for these close calls say what's going on is
I have a question. The
yeah.
slide here, is that a bladder cancer that we're seeing on this slide?
This is a really zoomed in mouse tumor.
Mouse tumor of bladder cancer
Bladder
mouse.
cancer. Yeah.
Mhm.
So you probably don't have your pointer, but you're seeing something that's like, that's it. It's identifiable to you. would you say, or
Would be-
like squiggly things, and some are white and some are really dark purple, but I, I'm not asking for a lesson in histology, but you're seeing something on here that's saying, oh, there's something here, and then you with AI you compare like a trillion millions of images
Mhm.
like this, and they come up with patterns. Is that correct?
Yeah,
Is that how it
exactly.
works?
Perfect, yeah.
Yeah.
Yeah, this looks a little chaotic. If you looked into normal urothelium in the bladder, it would look very well organized. But the thing about cancer is it doesn't cooperate according to the rules. So this is actually a very aggressive tumor.
Oh, okay.
Yeah.
Yeah, so gonna try to wrap it up real quick so everyone, if they have questions, can talk. But
So wait,
yeah.
Sam, where does the AI come from? Is it a computer program that you all have, or do you go to some AI thing in the sky? I mean, we're all like kind of really, we're hearing about ChatGPT and we're hearing about AI.
Mhm.
Where does AI live in terms of you're scanning all these slides, and how does that work?
So usually, go ahead.
Oh, go ahead.
No, go ahead, Stan.
Oh, I was just gonna say, we're kind of at the very beginning, we're collecting all the data so that, when this, we have enough data to be able to kind of be able to say, okay, this is this, this is that. And then, eventually we're going to be able to, start that.
Okay.
Yeah, so what Kai was talking about, so you use computer programming with really, really powerful computers, because as you said, Stephanie, this is a lot of data. This is a massive amount of data. So together with the kinds of computer programs that Kai and others are writing, you can teach the machine to do iterative learning. It can see the same thing over and over again in different slides, and it can start to learn patterns. And that repeated thousands and thousands and thousands of times is how the computer learns.
Well, what we learned today is, this has just been fantastic. Our brilliant young people working in obscurity, never seeing daylight, working with mice. Kai looked like he was in his basement, let him out every now and then.
He's actually in the office adjacent to the GBCI lab.
Okay.
We don't let him out.
What we're learning is without patients, none of this research can happen. And what we're learning, if I can do this intelligently, is you're looking for cells, you're looking at cells, at genetics, you're looking for clues, and you're looking for patterns and matches, and also observational studies. As Alina mentioned, you're seeing something that's leading to something, and why is that happening? So it must be kind of amazing to have all these questions. One thing probably leads to another all the time, and your curiosity is commendable. Please keep going. And I'm sure that, so a question for you. You're working on minuscule microscopic little things. Do you feel that you're making progress? And that's like a group question. And how do you feel about being part of the solution for bladder and upper tract cancers? So you can answer one or you or all of you, whatever you want.
Yeah, so I mean, I think, when we think about microscope slides and, very small things, yes, it is very microscopic, it is very small, but yeah, the tumors of cancer are extremely small sometimes. It's not necessarily about, the size, but the kind of the impact of what's going on. So yeah, it's important to kind of look at every single aspect of what's going on, and knowing what's important and what's not. And that's the hard part, right? Trying to figure out all that.
Well,
So
what you do is meaningful and rewarding. And I will say it's sort of a golden age for bladder cancer research. And you're very much part of it. The GBCI is now 10 years old, and I don't think there was a lab 10 years ago doing this kind of work. And it happens in other places too, clearly. I mean, there's all these amazing scientists all over the world who are working on this, but We feel that you all are very, very special, and we thank you for the work that you do. Because ultimately there's a person, as she so beautifully said, there's a person who has this and that you're trying to solve that problem for. So thank you for that. David, I'll let you,
Sure. So Karen has a question. Can a patient request pictures of their pathology slides? Some patients need a visual. I think you should definitely request it. I mean, again, if you're at Hopkins, we can make that happen for sure. But yeah, this kind of scanning technology is present in most academic medical centers now. So go ahead and ask.
I do have a question. Is it common outside of research to sequence tumors now? Is that sort of a standard that's come along? I know there's
Yes.
insurance issues and all that. And
Yes.
if you're not getting sequenced at your local hospital or clinic, can you ask for that? And how important is that?
So there are a lot of academic urologists who are sequencing their patients at the time of cystectomy
Okay.
because they want to have the sequencing done so that the patient has access to liquid biopsies. Because one of the companies that's very familiar to people is called Natera, and they make a designer assay based on the sequence of your tumor. But that can take a long time. So, Noah doesn't want his patients with advanced metastatic disease waiting 3 or 4 weeks for the test to be developed. So if they sequence the patient at the time of cystectomy, then they have the designer test in hand. So I know several of our colleagues across the country are doing this now.
Is
I
that
don't
paid
know.
by insurance yet, or in certain cases?
Yes, and if it's not, then Natera's told us that they'll eat the cost. But yes, it should be covered by insurance because of the.
By the way, if anyone listening has any questions or after the fact questions, we are bladdercancer@jhmi.edu, Johns Hopkins Medical Institution.edu. Just send us the question and we'll answer it. A wonderful lady, Anne, asked a question the last time, and we tracked you down. We found your email and we tried to answer it offline. But please, if you have a question, send us your email address so we can respond more quickly without going through all the data to find you. But we answer every question that comes along, and especially the question that David just answered about you can access sequencing even if it's maybe not offered in your sort of local community. It's important information to have, clearly. So sorry, I just walked all
That's
over you.
great. That's great. And again, if you ever are in the neighborhood and you want to come on by the laboratory, we could happily give you a tour and promise that we'll have the chef up and running so you can see that movie that Stan made. So
Thank you, everyone.
yeah,
And then
thanks
our
everyone.
next, David, our next one is on what, March 19th?
March 19th, yeah, with our own Noah Hahn and Max Cates, right?
Yeah, featuring their new multidisciplinary clinic. They're working together in the same clinic for the important sector who have high-grade, non-muscle invasive bladder cancer. That's a
Correct.
really important area. We don't want you to like escape and go over the fence to muscle invasive. And they have a unique clinic and work together to sort of give you everything, every tool in the toolbox, many of which you're developing right now for the future to stop the growth or stop the metastasis or shrink the tumor. I love what Alina says, melt it away. Let's do that.
Melt the tumor.
That's on March 19th. And anyone who wants, we'll have two our urologists and oncologists and ask their questions there. So thank you all for coming tonight. And
Thank you.
thank you all to our beloved and just cherished lab helpers and PhD and MDs from all over the world. We're like the United Nations. We're so lucky to have you all. Thank you.
Take care, everyone.
Great night.
Bye-bye.
Happy Valentine's Day.
Happy
Bye-bye.
Valentine's Day.